Section 3 of The Psychic Life of Micro-Organisms by Alfred Binet. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or volunteer, please visit LibriVox.org. Recorded by Leon Harvey. Chapter 3 The Psychology of Nutrition. 3. The mode of nutrition among microorganisms is not uniform, a fact which ought not to appear remarkable when we bear in mind that this immense group is made up of all manner of heterogeneous beings that have nothing in common save the microscopic littleness of their bodies and the simplicity of their structure. 3 main types of nutrition may be briefly distinguished. 1. Vegetable nutrition, or according to Bushley's expression, holophytic. This is the method of nutrition among animal or vegetable cellules that contain chlorophyll and that nourish themselves by forming organic nutriment from ingredients taken from the surrounding medium. It is hardly necessary to call to mind that the function of chlorophyll is that of nutrition and not of respiration. This phenomenon was formerly termed the diurnal respiration of plants. The expression involves several mistakes, enough to say that vegetables respire as animals do, by uniting with oxygen, and that respiration continues the same both day and night. The function of chlorophyll is by no means respiration. Its office is to decompose the carbonic acid gas of the air and to seize the carbon, which serves the plant in forming tertiary or quaternary substances. This chemical work is performed by all chlorophyll organisms when influenced by the radiation of light. Chlorophyll does not belong exclusively to the vegetable kingdom. A large number of animal microorganisms are colored green by this pigment. They are met with principally in the important group of flagellates. The assimilative organs, which are likewise found in all green plants, bear the name of chloromatophores. They have lately formed the subject of interesting investigations. The chromatophores are small bodies of protoplasm, which are distinguished from protoplasm in general by their having assumed an individual structure. These little bodies, which in the vegetables are called leucites, have a granular and reticulate structure. They are impregnated with a coloring substance, at times green, at times yellow, and at times brown, as the case may be. In fact, several coloring substances are present, which by intermixture in different proportions form colors of many varieties. The best known, after green chlorophyll, is yellow chlorophyll or diatomin. The latter coloring substance can be absorbed by alcohol. The euglenoideae, the chlamydomatidinae, and the vulvocinae exhibit enormous chromatophores. In the case of euglenoideae, the chromatophores are formed of small discoid plates that are situated directly under the cuticle so that the light can act upon them. See Figure 4. In certain species of flagellata, they are exhibited under the cuticle in the form of 2 large plates which envelop the protoplasm like a cuirass formed of 2 pieces. The chlamydomatidinae and the vulvocinae have green chromatophores, disc-shaped and very small. In the center of the chromatophore, a small bright space is observed which, when formerly thought to be filled with chromatophyll, in reality is a minute solid globule which shows an extremely close analogy with the substance composing nuclei, or nuclein. It exhibits the same chemical reactions. It actively absorbs coloring matter and grows extremely brilliant when treated with acids. Schmitz gives this little body the name of paranoid from pairin, nucleus. It is around the paranoid, and probably through its action, that starch forms, is deposited in grains or reunites in a ring about the paranoid, a fact easily ascertained by colouring them with iodine. Production of starch has also been observed in the colourless flagellates, as for instance in the Polytoma uvelia. These latter do not have chromatophores, but Kunstler, and after him Fish, have noticed that every grain of starch is attached to a small mass of colourless protoplasm, which is a focus of formation for the grains. This is precisely what happens in vegetable organisms where colourless starch leucites are found. This little mass of protoplasm always faces the helium of the starch grain. As the function of the chromatophores is exercised only when subjected to the influence of light, it follows that green microorganisms must have light in order to nourish themselves. A quite remarkable fact may be adduced in this connection. On examining the kingdom of protozoans as a whole, it will be seen that a striking coincidence exists between the presence of the eye and the presence of the chlorophyll pigment. Organisms having an ocular spot are in most cases provided with the chlorophyll pigment, or in other words, nourish themselves as plants do by generating starch through the action of light. This fact proves that sensibility to light is in some manner dependent upon the chlorophyll function. If flagellates possessing chromatophores, that is, organs generating starch, have ocular spots at the same time, it is because these rudimentary eyes enable them to find their way towards the light, which is the necessary agent of chlorophyll action. Accordingly, all microorganisms having eyes nourish themselves as plants do. In their case, the object of the eye is to direct the performance of a vegetable function. It is interesting to note in this connection that the euglena might nourish themselves as animals do, for they have a mouth and a digestive apparatus. The buccal, or oral aperture, opens in the anterior end, at the base of the flagellum, and is connected with a short gullet or esophagus. See Figure 6, the mouth and gullet of the euglena. Nevertheless, the euglena is never seen using its mouth for swallowing elementary particles. A quite curious problem is involved here, if it is true, as has been claimed, that it is the function that makes the organ. How do we explain the existence, and especially the genesis of this digestive apparatus which performs no function? In the presence of chromatophores that prevents certain flagellates from feeding like animals, so much so in fact that the digestive apparatus performs its function in flagellates which have no chromatophores and are not provided with chlorophyll pigment, an instance of which is seen in the Paraneema. The Paraneema is, further, an exceedingly voracious animal. We must note, also, that the Paraneema does not exhibit ocular spots like the green euglena, and moreover, it has no need of such, since it does not have to seek the light to generate starch. All these phenomena are interdependent. The influence exerted by light upon the green organisms of both kingdoms has been ascertained by different scientists. Light at a certain degree of intensity attracts them, and at a greater degree repels them. Some years ago, M. Strasburger conducted a series of connected experiments upon the movements of green spores towards light. It was observed here that the grains of pigment in the interior of the cellulose, when under the influence of solar radiations, executed movements and set outward in all directions. 2. Nutrition by endosmosis, or saprophytic. The organism nourishes itself by absorbing through the whole surface of its body liquids containing the products of vegetable or animal decomposition. Saprophytic beings are found in putrid waters or in infusions. This matter of nutrition may be considered, from the point of view which now engages us, as the most simple of all. It probably allows of a search for food, but it is certain that no movements are involved which are designed to draw the food into any possible digestive apparatus. 3. There is now a last mode of nutrition, of which we shall treat in minute detail, namely animal nutrition, where the micro-organism seizes solid elementary particles and nourishes itself after the fashion of an animal, whether it be by means of a permanent mouth, or by means of an adventitious one, improvised at the moment of need. This manner of nutrition is the process employed by higher animals. Among the lower organisms, it is met with in most of the infusoria, in the sarcodines, in many of the mastigophores, and in others. Respecting the microorganisms belonging to the vegetable kingdom, we find nutrition by endosmosis and chlorophyll nutrition. The protophytes never possess a mouth and never absorb solid foods. Animal nutrition requires very remarkable psychological faculties in the organism practicing it. These manifestations of psychic life, the progressive complexity of which we intend to trace in starting from the simplest protozoic forms and arriving at the higher, prove that these animalcula are endowed with memory and volition. We shall group our remarks under the two following heads: A. The choice of food, and B. The movements necessary for the prehension of food. The microorganisms do not nourish themselves indiscriminately, nor do they feed blindly upon every substance that chances in their way. Also, when they ingest food through some point or other of their bodies, they understand perfectly how to make a choice of the particles they wish to absorb. This choice is sometimes quite well defined, for there are species which feed exclusively upon particular foods. Thus there are herbivoria infusoria and carnivorous infusoria. Among the herbivorous ones may be classed the chelidons, which feed upon small algae, diatomaceae, and oscularia. Parameciea live principally upon bacteria. The leucophores is a specimen of the carnivorous class. It devours even the smallest animals of its own kind. The Certostomum leucus eats everything, as do the rotiferous. Though the fact of an exercise of choice in taking food is settled beyond question, yet the interpretation of this phenomenon is a matter of much uncertainty. Some writers, as Charlton Bastian, for instance, explain this choice of food as an affirmity of chemical composition existing between the organism and the nutriment. This idea does not lead to anything. Others compare the discrimination made by the proto-organisms between objects presented to it and the action of a magnet, which in some way selects particles of iron that have been mixed with particles of other substances. The latter interpretation is an evidence of the tendency evinced by some naturalists of endeavouring to identify the attributes of living organic matter with the pseudo-chemical properties of the mineral kingdom. In our opinion, the only question demanding consideration is whether the choice of food, in the case of proto-organisms, does or does not result from a psychical operation, similar, for example, to that which takes place in higher organisms. We have received a noteworthy communication from M.E. Morpas upon this subject, which tends to establish that the choice of food is not the result of individual taste in the microorganisms, but is determined by the organic structure of the buccal apparatus, which does not allow them to receive other forms of nutriment. We must closely examine, therefore, the mechanism for prehension of food. The following is what occurs when the amoeba, in its rampant course, happens to meet a foreign body. In the first place, if the foreign particle is not a nutritive substance, if it be gravel, for instance, the amoeba does not ingest it. It thrusts it back with its pseudopodia. This little performance is very significant, for it proves, as we have already said, that this microscopic cellule, in some manner or other, knows how to choose and distinguish elementary substances from inert particles of sand. If the foreign substance can serve as nutriment, the amoeba engulfs it by a very simple process. Under the influence of the irritation caused by the foreign particle, the soft and viscous protoplasm of the amoeba projects itself forward and spreads about the elementary particle somewhat as an ocean wave curves and breaks upon the beach. To carry out the simile that so well represents the process, this wave of protoplasm retreats, carrying with it the foreign body which it has encompassed. It is in this manner that the food is enveloped and introduced into the protoplasm. There it is digested and assimilated, disappearing slowly. There are cellules found in the inner intestinal walls of lower animals, which effect the prehension of solid foods in the same manner as the amoeba cellule. They are called phagocytes. This mode of prehension is beyond contradiction, the most simple imaginable, for the prehensile organ is not as yet differentiated. Every part of the protoplasm may be made to serve as a digestive cavity in enveloping the foreign substance. From the special standpoint of prehension of food, we may place the actinophores sol above the amoeba. This animalcule is a small microscopic alenazolarian, abounding in freshwater ooze. It carries out long, slender filamentous basopodia from every part of its body. When its prey or any elementary substance gets into the midst of this mass of filaments, the filament affected quickly draws back, carrying the nutritive matter with it towards the body proper of the actinophores. In other instances, the filaments, anastomosing themselves, form a sort of envelope about the prey. At the instant the substance comes within a short distance of the cellule, the part of the protoplasm composing the mass projects itself forwards, and encompasses the food, which is carried back and enveloped in the midst of the protoplasm by a process analogous to that seen in amoeba. In the case of the actinophores, any part of the body could serve as a way of entry for food, that is to say, could act the part of a mouth. To use the expression of W. Saville-Kant, it is a pantostomate being. In other species of higher organization, this mode of alimentation is rendered impossible by the cuticle which encompasses the body. The formation of a cuticle impervious to solid foods creates the necessity of a buccal orifice through which food may enter into the interior of the protoplasm. A curious graduation in these phenomena is noticed here. Thus there are organisms destitute of a permanent and pre-existing mouth. Their mouth is improvised as the occasion demands, is adventitious, so to say, and the reason that these organisms are ranked higher than the preceding ones is that the mouth is invariably formed in the same place. In this connection we may examine a small flagellate infusory which abounds in impure waters, the Monas vulgaris. It carries a long flagellum attached to its anterior extremity, which, when not in motion, is coiled up against the body. At the base of the flagellum, the protoplasm projects a pellucid substance in the shape of a lip. The protuberance is hollow, containing a vacuole filled with liquid. Zientkowski has described how these different organs act. The bacteria and micrococcus, which constitute the food of the monas, are pulled into the latter's neighborhood by strokes of the flagellum. At that instant, the animal becomes conscious of the proximity of these other bodies, for the protuberance which lies at the base of the flagellum extends towards the corpuscle, envelopes it in its own substance, and pulls it back into the interior of the monad's body. The Butchley has made an analogous observation with the Ochomonas termo. The prehension of food comprehends here 3 phases, in 2 of which the organism manifests psychical activity. First, attraction of food by means of the flagellum. Second, formation of the vesicle, which extends towards and envelopes the food when the latter has come near. Third, the absorption of the food. The Actinitae are organisms that move about very little. They frequently remain fixed to a pedicle their whole life long. They have no cilia, but exhibit radiating prolongations, more or less numerous, and sparse or grouped in tufts, as the case may be. These filaments are suckers, provided at one end with a small air hole. When a thoughtless infusatory swims into the territory of an Actinita, the latter arrests it by means of its stout filaments and fastens upon the former's body the cup-shaped extremities of its suckers, which make a vacuum. The protoplasm of the ciliate, thus captured, slips slowly through the suckers as through tubes, and is gathered together in the interior of the Actinita in the form of small drops. In the Actinitae, accordingly, particular organs are adapted to the prehension and absorption of food. Corresponding to the greater complexity of physical action, the psychical process necessary for the act of prehension has likewise become more complicated than in the case with the amoeba. The Actinita is obliged to direct its sucker towards the infusory which is within its reach, and consequently the animal is obliged to determine the position of its prey. There are Actinitidae that exhibit prehensile organs more perfect than those just noticed. Such are the hemiophores. They have both sucker tentacles and prehensile tentacles. The latter are filaments which the animal throws about its victim, like a lasso, thus enveloping and rendering it motionless, while it proceeds to feed upon it by means of its suctorial apparatus. Now, do these Actinitidae show any preference of choice among the infusoria that chance to fall within reach of their tentacles? M. Maupas, who has made an especial study of these organisms, had at first admitted this preference and choice, but he afterwards rejected the notion. In 1885 he writes thus: "I find quite another explanation of the impunity with which the Collaborator can throw itself upon the terrible suckers of the Potopheres fixa. The stout shell with which this little infusory is enveloped serves it as a shield and guards it from the deadly grasp of the Actinitidae. The Actinitidae do not seize the collops because of any dislike of the latter, but because they are unable to seize them, and their inability results from the peculiar structure of the collops tegumentatory envelope. The Paramecia, which also escape unscathed, are similarly provided with a tegument of high resisting power, which serves them as a protection in this contingency. The Stylonychiae hisfurio, like all other Stylonychiae, has a very soft tegumentatory envelope. They are accordingly seized and devoured by the Actinitidae without difficulty. The detailed knowledge of the differences of structure in the tegumentatory envelopes has caused me to abandon the idea of a preference or dislike in the choice of those victims which serve as food for the Actinitidae. Of the prey that passes by, they catch what they can, and not what they want to." In a large number of species, the prehension of food is preceded by another stage, the search for food, and in the case of living prey, by its capture. We shall not investigate these phenomena among all the protozoa, but shall direct our attention especially to the ciliated infusoria. Their habits are a remarkable study. If a drop of water containing infusoria be placed under the microscope, organisms are seen swimming rapidly about and traversing the liquid medium in which they are in every direction. Their movements are not simple. The infusoria guides itself while swimming about. It avoids obstacles, often undertakes to force them aside. Its movements seem to be designed to effect an end, which, in most instances, is the search for food. It approaches certain particles suspended in the liquid. It fills them with its cilia. It goes away and returns, all the while describing a zigzag course similar to the paths of captive fish in aquariums. This latter comparison naturally occurs to the mind. In short, the act of locomotion, as seen in detached infusoria, exhibits all the marks of voluntary movement. It might also be mentioned that every species manifests its personality in its mode of locomotion. Thus, as a rule, the Actinotrichia saccans, when placed in a preparation where it finds itself at ease, remains for a few moments perfectly immovable. Then of a sudden it dashes forward with the rapidity of lightning and disappears from the field of vision. For a time it darts about to the right and to the left, and then once more assumes its state of immobility. It can move with the greatest agility through masses of debris, in the midst of which, bending and twisting, it slips about with wonderful nimbleness. The Legones crassicoles, on the other hand, moves along at a pace quite constant and uniform, neither slow nor rapid. It searches about among algae and fragmentary particles. The Peritroma seme moves slowly. It runs lazily over the algae where it seeks its nutriment, and does not stray from them to venture into the open water. Concerning the prehension of foods and the search for nutriment on the part of the ciliates, we can do no better than to quote entire a note which M.E. Morpus has been pleased to send us upon the subject. We had put to him two questions. First, do the ciliates hunt their food? Second, while in quest of live prey, do the ciliates called hunters make an actual hunt, involving the espial of prey from a distance and the voluntary pursuit of the same in the circuitous paths they follow? M.E. Morpus, after having once more had recourse to observation, briefly recapitulates his opinion in the following lines. From the standpoint of prehension of food, the ciliates may be divided into two great groups. 1. Ciliates with alimentary vorticles. 2. Hunter ciliates. In the first group, the mouth is always held wide open, and among the nutritive particles, which the current of the vortex keeps constantly sucking in, we may at will cause other absolutely inert and indigestible particles to take the same course. For instance, such subjects as granules of carmine, indigo, and rice starch. These granules, totally unfit for nutritive purposes, pass through the body of the ciliates along with the genuine nutriment, and are finally cast out intact with the excrement. I think, therefore, we may affirm that the species having alimentary vorticles exercise no real choice in selecting their foods, and that they absorb indiscriminately all the corpuscles which by reason of their form and density admit of being seized and drawn into the alimentary whirlpool. In the case of the hunter ciliates proper, the mouth is constantly closed. The act of absorbing each object captured is accomplished by a process of deglutition, comparable in every phase to the like processes in higher animals. Furthermore, these species feed only upon living prey, which they capture and entrammel by means of their trichocysts. Vid Archives de Zoologie, Volume 1, 1883, page 607, and ff. By this very act they exercise a choice in the selection of food. But this manifestation of choice is not, in my opinion, the result of preference, or of individual taste, but is the consequence of the peculiar construction of their buccal apparatus, which does not enable them to take other and different nourishment. These hunter infusoria are constantly running about in quest of prey, but this constant pursuit is not directed towards one object any more than another. They move rapidly hither and thither, changing their direction every moment, with the part of the body bearing the battery of trichocysts held in advance. When chance has brought them in contact with the victim, they let fly their darts and crush it. At this point of the action, they go through certain maneuvers that are prompted by a guiding will. It very seldom happens that the shattered victim remains motionless after direct collision with the mouth of its assailant. The hunter accordingly slowly makes his way about the scene of action, turning both right and left in search of his lifeless prey. This search lasts a minute at the most, after which, if not successful in finding his victim, he starts off once more to the chase and resumes his irregular and roving course. These hunters have, in my opinion, no sensory organ whereby they are enabled to determine the presence of prey at a distance. It is only by unceasing and untiring peregrinations, both day and night, that they succeed in providing themselves with sustenance. When prey abounds, the collisions are frequent, their quest profitable, their sustenance easy. When scarce, the encounters are correspondingly less frequent, the animal fasts and keeps his Lent. The Leginus crassicolis, accordingly, never sees its victim from a distance, and in no case directs its movement more towards one object of prey than towards another. It roams about at random, now to the right and now to the left, impelled merely by its predatory instinct, an instinct developed by its peculiar organic construction, which dooms it to this incessant vagrancy to satisfy the requirements of alimentation. The vorticell infusoria, when in a medium abounding in food, are almost entirely sedentary in their habits, only making slight changes of position. But if they are placed in a medium afforded by little nutritive material, they become as migratory as the hunters, and are seen to race about in all directions searching for more abundant nutriment. It is hard to find a more perfect illustration of the influence exerted by the conditions of a medium upon the habits and customs of animals. The Luciferus patelia is a type distinctly carnivorous and possessed of an extremely voracious appetite, a fact which explains its power of multiplication, one of the greatest I have studied. With a temperature of 25 degrees in my laboratory, I have recently seen it separate by fission 7 times in 24 hours. That is to say, a single individual produces from itself just 128 others in that time. In constant pursuit of its prey, it seizes its victims by the 2 stout vibratile lips with which its mouth is armed, and swallows them alive and whole. The victims may be seen struggling and tossing about for a time with the interior of the Lucifer's body, and afterwards to expire slowly under the action of the digestive juices of the vacuole in which they have been enclosed. Placed in a medium well stocked with small ciliates, the lucifers have their bodies constantly crammed with victims, swallowed in the manner above described. Like the inner hunter ciliates, the lucifers does not espy its victims from a distance and does not guide itself towards them. It simply darts about from right to left, every moment changing its direction. It thus increases its chances of coming in collision with its prey, and every time that one of its unfortunate victims falls in contact with its vibratile lips, it is seized, irresistibly drawn towards the mouth, and swallowed within less than a tenth of a minute. Certain hunter infusoria have methods of pursuit and capture which deserve to be examined separately. Claparede and Lachmann, in their excellent work upon infusoria and rhizopods, have minutely described the manner in which a large infusori, the Amphileptus meleagris, attacks the Epistelis plicatilis. The epistelis are colonizing vorticles of which certain individual members attain a size of not less than 0.21 mm. The epistelis form absorbent groups, the ramifications of which are quite regularly dichotomous. These ramifications all grow at exactly the same rate, and the individual branches all rise to the same height, representing what is called, in botany, a corymborous inflorescence. We were observing one day, says Claparede, in the hope of seeing what would come of the maneuver, an Amphileptus, which was slowly creeping up a colony of Epistelis. The way in which it approached the vorticels, feeding them, so to speak, and partially enclosing them in its pliable body, already seemed suspicious. At last it made a direct attack upon one of them, by fastening itself upon the upper part of its body. It opens its huge mouth, which is never to be seen except when the animal is eating, and slipped over the epistelis like the finger of a glove being drawn upon a finger of the hand. We saw the size of the buccal aperture, which are capable of being dilated in a truly astonishing manner, slip slowly over the peristome and upon the body of its prey, and then draw together about the point where it was made fast to the pedicle. The cilia covering the body of the Amphileptus began to shake with that peculiar motion which is always noticed when a ciliated infusori secretes a cyst. At expiration of a moment or so, a fine line was seen to appear around the whole body, which continued to spread so as soon to form the cyst. This might be called a cyst of digestion. The phenomenon as a whole is quite simple. An Amphileptis approaches an epistelis, devours it, and insists itself upon the spot, the victim being still attached to its pedicle. It then endeavours to wrench the epistelis from its point of attachment by twisting. It turns on its axis from left to right, and then from right to left, successively. When it has succeeded, it continues its work of digestion, and occasionally divides in two within the cyst itself. During the last stage of digestion, it rests for a while, when it commences again to turn about in the cyst, evidently seeking to disengage itself. At the close of a certain number of hours, the cyst breaks. The Amphileptis issues forth and starts in quest of another victim. The hunter infusoria are frequently armed with trichocysts. Trichocysts are vertical filaments which serve the animalcula provided with them to disable or wound other microorganisms. A large number of infusoria, the Paramecia, the Ophrioglinae, etc., use their trichocysts as organs of defense. With other species, of which we shall speak more at length, the trichocysts are organs of offense. They are located either in the sides of the mouth or in parts adjacent thereto. This is the case with the lacrimaria, the duodenum, the encheles, the leginus, the loxophilium, and the amphileptis. These latter animalcula attack the live prey that constitutes their food in the following manner. They dash upon their victim and bury the trichocysts with which they are armed into its body. The victim is immediately brought to a halt, whereupon the hunter seizes it and swallows it. So when the Laginus elongatus intends to seize a victim that has fallen into its vortex and has thus been drawn into the neighborhood of its mouth, it throws itself swiftly forward. At the moment of contact, the hunted infusori becomes suddenly paralyzed and remains perfectly motionless. This paralysis is evidently caused by the trichocysts, which line the esophagus of the Laginus, and with which the latter has transpierced its prey at the moment it came in contact with its anterior extremity. In a higher stage of organization, the microzoon possessing a mouth changes its position in order to intercept its prey and give it chase. The Duodenum nasutum, Steen, are carnivorous infusori, and one of the most voracious of our fresh stagnant waters operates in a more complicated manner. It casts its trichocysts upon its victims from a distance. The importance of this instance induces us to stop here a moment. Figure 7 is displayed on the page. Duodenum nostrum. The duodenum, Figure 7, as regards the general shape of the body, may be compared to a diminutive cask, rounded off at one of the ends and terminated at the opposite extremity by an almost level surface, from the midst of which rises a conical projection, quite strongly marked. This projection is an organ of deglutition, swallowing. A longitudinal striation is noticed here, formed of minute solid rocks, of extreme tenuity and independent of the sides. These organs are the weapons used by the duodenum in attacking the live prey which constitutes its sole nourishment. Not only does it attack and devour animalcula almost as large as itself, but frequently it even seizes individuals of its own kind. In such cases it is always infusoria and never the roteroria. Although the latter often abound in waters which the duodenum inhabits, it appears moreover to have a marked predilection for certain species, and so it happens that the huge and inoffensive Paramecia aurelia is almost always its choice of preference among the animalcula that inhabit the same liquid. The prehension of food by the duodenum exhibits interesting aspects, which have not as yet been observed in any other infusori. M. Balbiani, in his first observations, had often been surprised at seeing animalcula that the duodenum had passed by without touching suddenly stop as if violently paralyzed, whereupon our carnivorous specimen straightway approached and seized them with seeming facility. More careful examinations of the duodenum's actions soon furnished the key to this enigma.
If while swiftly turning in the water, the duodenum happens into the neighbourhood of an animalculum, say a paramecium, which it is going to capture, it begins by casting at it a quantity of bacillatory corpuscles which constitute its pharyngeal armature. Paramecium immediately stops swimming, and shows no other sign of vitality than feebly to beat the water with its vibratile cilia. On every side of it the darts lie scattered that were used to strike it. Its enemy then approaches and quickly thrusts forth from its mouth an organ shaped like a tongue, relatively long and resembling a transparent cylindrical rod. The free extended extremity of this rod it fastens upon some part of the paramecium's body. The latter is then gradually brought near by the recession of this tongue-shaped organ towards the buccal aperture of the duodenum, which opens wide, assuming the shape of a vast funnel in which the prey is swallowed up. Up to this point we have paid little attention to movements of defense and of flight. Upon this subject a few words will suffice. When vorticils are alarmed, they are seen to contract forcibly their pedicle, which, in a state of rest, stays extended. Infusoria are placed in a preparation where they are at their ease, swim quietly about. If any sharp extradiction disturbs them, they accelerate their pace. Those armed with a rigid bristle at the posterior extremity rush precipitately onward whenever another infusory chances to touch that tactile appendage. The unaggressive paramecia, when attacked, endeavour to escape, but are also able to defend themselves by means of the trichocysts with which their ectosac is armed. End of section 3.